I would like to thank the organizers and everyone for the opportunity to present today. I am in Puerto Rico right now. Unfortunately, I couldn't come to Mexico due to a family emergency, but I'm glad that I can be here via Zoom. So as you can see, the title of my talk is Identification of Cancer Drug Gene Resistant Network Modules by Analysis of Genome-Wide Expression and Drug Activity Correlations in Cancer Cells. As you'll probably know, cancer is one of the top 10 killer diseases in the world, and the latest available stats from the Global Cancer Observatory indicate that there are about 19 new million cases that were diagnosed and close to 10 million deaths in 2020. You can see that breast, lung, and colorectal cancer lead the way in diagnosis, while lung, colorectal, and liver top the mortality rates. There we go. In their seminal paper, Hanahan and Weinberg outlined the biological characteristics of human cancer, which included initially sustained proliferative signaling, resistance to apoptosis, evading growth suppressors, inducing angiogenesis, enabling replicative immortality, and invasion and metastasis. They subsequently added additional hallmarks: reprogramming of energy metabolism, evading immune destruction, genomic instability, and tumor promoting um, inflammation as enabling characteristics of the process of tumorigenesis. Now, these are all well known as the famous hallmarks of cancer. There are two forms of drug resistance in cancer: acquired and innate, also known as intrinsic resistance. Acquired resistance usually develops during treatment, but innate resistance is a possibility even before treatment. Acquired resistance is developed during the treatment and is due to mutations or adaptive responses to treatment. Drug resistance is most often the result of therapeutic selection of a subset of tumor cells, uh, which tend to have a high degree of molecular heterogeneity, as somebody mentioned already over here. Mechanisms of drug resistance will include, for example, drug efflux, drug inactivation, modifications in drug targets, and inhibition of cell death. And there's a lot of study going on as to what are the genes involved in these processes and how they collaborate with one another and the differences that there are between the different cancer types. Now, you can see that we are dealing with several biological hallmarks that enable cancer progression and multiple mechanisms of drug resistance that make treatment difficult. Therefore, we must continue to research molecular intricacies of cancer and seek new treatment therapies. So, what we're doing is using bioinformatic techniques and pharmacogenomic approaches to look for new plausible drug therapies to identify new molecular targets to help better understand and curb, the ravages of cancer. Uh, Everyone knows in here that cancer is a complex disease where multiple genetic changes rewire cellular networks during carcinogenesis. And this indicates that cancer drug therapy needs the implementation of network-driven studies to reveal multiple interactions between cancer genes and drugs. Now, what do we use? We use CellMiner, Cosmic, and GDSC databases uh, to gather the data and to do the analysis in this research. Data about the genome-wide expression of human protein coding genes in the NCI60 cancer cell lines were downloaded from CellMiner. And this is a pharmacogenomic resource that reports the activity of about 25,000 compounds right now. However, I must point out that they do not include monoclonal antibodies. So that's a deficiency in the database. The expression data from the human genes corresponded to mRNA normalized signal for each gene, and it was then converted to the average Z-score. The selection of cancer genes used in our studies was then taken from the cancer gene census data set in Cosmic. And from there, we got 733 cancer-related genes common between the CGC and CellMiner that we analyzed using the GDSC to corroborate the results. Once we filter significant negative correlations, which indicate resistance to drug therapy, and FDA-approved drugs, we identified 1,552 resistant pairs between 137 drugs and 374 genes. Heat maps were then generated to establish resistance cluster by cell lines of different tissues, as well as gene drug bipeptide resistant networks. And the results show the identification of known resistant gene drug pairs and some plausible new ones. So that's really exciting. So over here, you have a heat map of negative correlations. These heat maps were produced to better visualize the data because we have a lot of data. And over here in the bottom axis, we have all the NCI60 cell lines. The red color indicates a gene with significant positive expression and negative correlation, while the blue color indicates a gene with negative expression or that correlation is not significant. And the intermediate colors are genes that don't reach that significance threshold of a z-score greater than plus or minus 1. Here we have a schematic view of the network corresponding to the 1,552 resistant pairs between 374 genes and 137 FDA-approved anti-cancer drugs. The colors, I know you may not be able to see them very well right now, but they identify the drugs in red or pink, depending on your monitor. Oncogenes are purple or lilac, and tumor suppressor genes are green. Anything that is gray is that we don't know yet how it behaves in cancer, but that gene is involved in cancer. So we're exploring this network and performing enrichment to unveil the information contained within in order to identify new plausible resistant gene cancer FDA-approved drug pairs, as well as genes that may be involved in multi-drug resistance, also known as MDR. Over here, we have a subset of the heat maps, but this is for colon cancer cell lines specifically. So we have 1, 2, 3, 4, 5, 6, 7 colon cancer cell lines. And you can see that there are several significant clusters.
Monica, sorry to interrupt you, Monica. Your mic appears to have gotten muted on us. You can just tab back and click the audio.
Oh my!
Here we go. We're good. We're all set. Thanks.
Okay, so I need to go back to this slide here, or the previous one.
This one. Thanks.
This one. Okay, gotcha. Oh, thank you for letting me know. So as I was saying, this heat map shows the 6 or so colon cancer cell lines, and you can see that we have several clusters that point to drug resistance. Some of these we corroborated in the literature. Others seem to be new plausible gene drug pairs that we're still exploring. And over here, and I'm keeping track of time here so I don't go over. This subset over here is the bipeptide network for cancer, colon cancer specifically, and we see 159 resistant pairs between 56 drugs and 17 genes. CTNND1, for example, is a key regulator of cell-cell adhesion, and this one has the most drugs with negative correlations at 34, followed by transcription factors KLF4, and over here TCF7L2. They each have about 17 drugs with which they have resistance. And then we have ERBB2 over here with 13 significant negative correlations that point to drug resistance. Now, highlighted in these 4 boxes, as a matter of example, we have CTNND1, ERBB2, FOXO1, and KLF4. Now, these are known genes, to be involved in hallmark processes and poor prognosis, and some of them have also been shown to be involved in drug resistance. So we're still analyzing this network to try and uncover, various modules and new information. Because a good problem that we have right now is that we have a ton of data. So we're very lucky with the results that we got this summer, that we need to continue the in-depth analysis to, corroborate, these negative correlations that they indeed point to drug resistance and multiple drug resistance. We need to continue to determine the implications of negative correlations observed, determine protein targets that induce drug resistance, because there's still a lot of unknowns in these cancers. And we also need to finish an in-depth analysis of compounds not approved by the FDA for cancer treatment to propose plausible treatment candidates. Now, I would like to thank from the bottom of my heart Dr. Javier de las Rivas for welcoming me once again to his lab this summer for research. Alberto Berral Gonzalez, who's a very talented PhD candidate student doing amazing work. Jose Manuel Sanchez for statistical support, Diego Alonso for network support, and really everybody in the lab. They have been great. Absolutely, we need to, of course, acknowledge the different funding sources that make this work possible, and there are several of them. And with that, I think I made it just in time, just in the nick of time, for a question or two. So thank you for your attention, and I'll be happy to take any comments or questions that you have, or send me an email.
Thanks a lot, Monica. Any question for Monica?
Hey, Monica, really nice talk. Thank you. I have a question. Maybe it's a bit dumb, but I was wondering if you can detect already the resistance signals, like the genes that are correlating with resistance to drugs, not only in the tumor cells, but in the somatic cells of the patients. Could that already indicate that maybe we are prone not to be able to take some medication from the start?
That's actually a very good point. And in order to do that, we need to add an additional data set. One of the problems that we have right now is that the way data is reported is not standardized. So we first need to figure out how to level that out in order to make the appropriate uh, analysis and implications. But it's actually a very good point. And in theory, it is plausible because the data is out there. We just need to figure out how to make it make sense given the disparity of how the data is reported. But that's a very good point. Thank you for bringing it